Hi, I'm Bob Deans. I'm CSO at Synthego, and I'm here today to introduce you to the company and point you to a horizon we think is very, very exciting. Yeah, Synthego was founded by some SpaceX engineers, Paul and Michael Dabrowski, and really represent the innovative friction that forms when technology and engineering meet the life sciences sector. And so one of the real advantages Synthego had from the beginning was advice from some really renowned people. Jennifer Doudna, obviously, winning the Nobel Prize for the CRISPR enzyme discoveries. Matt Porteous, a really prominent engineer in genome engineer in our space. Bill Scarnes, Sue Andrew Whitty, have really formed the platform and basis for Synthego that's enabled us to support so much work in the CRISPR engineered cell space. What I'm going to do is turn you over to a video that's going to describe some of the core platform technology from Synthego and pick it up on the other side with a little bit more color and introduction to our future.
Advances in genomic technologies like next-generation sequencing have led to an explosion of data. Data that has unlocked a new level of understanding of human biology. Yet the reading of our genomes has created more questions than answers, while revealing ever-increasing layers of biological complexity. Unraveling this complexity requires new approaches to scientific research. Genome engineering technologies like CRISPR are enabling researchers to write back into genomes with unprecedented precision, while advances in machine learning and automation engineering are enabling the rapid and scalable production of engineered biology. At Synthego, we are a group of chemists, biologists, engineers, and bioinformaticians building scalable, vertically integrated full-stack factories. Factories we can use to engineer, model, and ultimately develop predictable biological systems. With access to our proprietary high-throughput platforms, scientists around the world can vastly accelerate their research applications, from basic drug discovery to disease modeling to the development of a new class of engineered biological therapeutics such as cellular and genetic therapies. At the true intersection of biology and engineering, Synthego is at the forefront of technological innovations. We are unlocking the vast potential of genome engineering and beginning to unravel the complexities of biology. Learn more about how you can unlock the potential of engineering biology with access to our automated platforms. Visit synthego.com now.
Yeah, I'm back, and what I'd like to do is introduce to you the technologies we're using to try to accelerate development in the cell and gene therapy space. And it's really based upon the platform technologies that were introduced to you in the video that you just saw. So it really begins with RNA synthesis, our mastery there. It enables people moving from left to right in the discovery chain to select and identify and define the right guides for gene editing. It really also incorporates automated and robotic workflows that help us design optimal engineered cell products, again, pointing towards GMP and clinical use. Our discovery platforms enable us to interrogate the engineered cell populations and identify potency pathways or target ID by discovery pathways that are highly informative moving into the clinic. And all of that is reinforced with a constant influx and evaluation of data using AI and machine learning. You know, we really feel we're significantly distinctive in this space in employing AI and machine learning to the discovery process. What I like to do now is point to several leverage points that are operating in our industry space and where we feel CRISPR-based discovery can be highly advantageous. And so on the right, really pointing to some metrics around the GMP cell therapy engineering space, obviously booming, with many examples of validated early technologies coming forward. And then on the discovery side on the left, it's really around translational genomics and how we incorporate clinical data from diseased patients or variants in an otherwise healthy population and really remarkably understand disease and redesign our approach to it. And so that's really what's behind Synthego putting so much effort into automating and utilizing an industrialized gene editing platform. It's really something we argue allows us to do disease modeling at scale and forms a very effective platform for agile cell therapy development. So here's a bright screen. What I want to do is break a little bit and show you some examples about how we're carrying things forward in the translational path. And what I want to do is really open up with a few slides that talk about the importance of doing things accurately and correctly. You know, it really opens up with the theme that our business is based on genetic diversity. We read it and rewrite it, and it's really what's enabling the development of this new class of medicines. And so what do we need to do to get there? We have to do it very pragmatically and safely. And so we don't have a precedent in front of us with a GMP development path highlighting the way. And so it's really dependent upon us to develop the analytics and the tools needed to be able to practice GMP editing in a safe way and hasten acceleration to the clinic. So what I'm doing here is showing you on the left some examples of our either our chemistry factory and our ability to synthesize very many guides very, very quickly. I'll show you an example in a second. And down below, the automated workflow that lets us iterate many cycles of a specific gene editing event. And so what you're seeing in each of these boxes are the number of cycles it's taken to optimize an editing efficiency from single digit to double digit, 90% knockouts, very highly efficient knock-ins. And what it's also showing you, if you look down on the right, is that we can do it with cells that are very difficult to edit. So looking at primary T cells as an endpoint, and the number of cycles we've gone through to build out a reliable and robust and highly efficient editing process. So the reason that's important is that clinical success is going to be driven around safety, and safety is going to be driven around the efficiency of hitting the desired target and not hitting a target that off target that potentially poses risk. And so what we're illustrating here, which is really a carry on from the previous slide, is our ability to use these automated workflows and move the efficiencies of our on targets to a very high level, here 7% up to an 80% level. In doing that, we also get statistical quantitation of our off targets linked to partial homology to guide and off targets that might be picked up as allelic drift as part of our cell handling process. What we know is that we can quantitatively, statistically understand our off targets and take that forward in a very tightly constructed package to the FDA for regulatory approval. And we've done that with several customers and clients several times. So I'm pointing now to some new technology we have that really augments the ability to control on-target access and limit off-target hits. And that has been our ability, we recently published this, our ability to generate guides that are sensitive to light. And we've really extended from our publication understanding that we can use light sensitive guides both to turn a reaction off as well as to turn a reaction on. So you can understand that where we're going is towards an ability to do very precise genome editing in a very precise temporal spatial pattern, whether we're looking at patterns in vivo for gene editing or whether we're looking at highly efficient ex vivo gene editing events. And so you can see we're building this capability forward to be highly effective in reducing regulatory risk and trying to drive accessible new genetic medicines into the clinic. So I'm going to step back a little and talk to you about our guide capability. Obviously, the RNA guides are key to making an RNP, which is key to doing editing under GMP or research conditions. And so Synthego has grown greatly in the ability to first introduce research scale guides at GMP for over a year now, and then rapidly amping up our capability in response to customer requests. There's a huge market opportunity here to provide GMP guides in a number of important industry segments. So one of those industry segments, obviously, is CAR T therapeutics, where the cost of utilizing lentivirus as a genome editing tool can bring very high expectations on reimbursement, very high cost of goods. And so, we all know those are technologies that are in the hundreds and hundreds of thousands of dollars to deliver. So just out of the box, gene editing reagents can come in, GMP reagents can come in magnitudes lower in cost in that treatment cycle. So it's a very attractive target to chase, an opportunity to chase. One of the ways we're also building out at Synthego is using our automation and engineering mindset to build automated GMP editing. And so we've built an automated workflow that's highly efficient compared to competitor devices in editing both primary and immortalized cell lines, very efficient in editing iPSC as well. And so it's very straightforward for us to now be able to begin to complete an offering of taking clients or customers from guide design through scale up under GMP and now through hardware and workflows automated that can deliver the edited cell product. So I'm going to switch gears now and talk a little bit for 4 slides or so about our disease modeling capability. And it's really driven around those same principles of being able to make RNA in factories and do automated robotic workflows that allow us to succeed in these cases. What I'm going to do is give you a couple of examples of 3 kind of libraries where we've had very high throughput and very compelling data coming back out. So one of the obvious uses of genome editing is to understand single nucleotide variants within a gene and to be able to link genotype to disease causation or to pathways that are linked to disease causation. So studying and linking genetics to disease is a very important part of our library offering. So one of the other things that is very valuable these days is to understand where regulatory mutations in the form of enhancers or promoters may actually be very relevant to disease and to be able to identify and screen for those. So synthetic lethality type screens that can associate several genes in a pathway that can be druggable to prevent transformation is an example of the use of that kind of library. And then something I'll show you in a second, which has been really fun, is the ability to quickly identify targets for a cell or gene therapy by getting assay-ready cells out in knockout cell pools. And so that's what's actually represented on this slide, which is a very productive collaboration we've had with Dr. Nevin Krogan at UCSF, at the Gladstone Institute at UCSF. And what Nevin had done that we had observed is he had studied the way that COVID proteins interact with the human genome. And he had done that by affinity purifying complexes between COVID and human proteins, identifying the human targets, and then introduced us to the opportunity to provide a screening service. The screening service was to knock out individual human proteins known to be required for COVID replication, and to do that in test cell lines that could be rapidly screened in infectivity assays. And so we did that. The data set that you see on the bottom involved our making over 1,000 guides in 3 days, constructing over 300 knockout libraries in 3 different cell lines, distributing those in 2 weeks to New York, Atlanta, and Paris in a screen-ready format, and getting knockout targets back in a 3-week period, so like that. So the work went on to progress over that, from the time that we began that to a 9-month period where findings from that have actually led to repurposed drugs that are out and effective in limiting COVID infectivity in phase 3 studies. So rapid discovery engine in that way. One of the other examples I wanted to show you is a series of iPSC libraries in which we introduced complex single nucleotide variants. And we did those to create a panel of wild type, homozygous mutant, or heterozygotic mutant clones that could be studied for disease causation in Parkinson's, Alzheimer's, and related chronic neurodegenerative disease. So this is something we were contracted to do with the NIH, and we did in the background of the COLF 2.1 iPSC line from Jackson Labs. You can see that in a year, we went from understanding the launch of the project in May to constructing over 250 different variants on target, and then being able to send those out to the consortia for testing in that scope of 7 to 8 months. So really productive project for us. I'm going to go ahead and close this section by stating something that's obvious, that with this power of genome engineering and the ability to apply an automated and robotic workflow, we can really look at deconvoluting very complex disease. We can associate genotype to phenotype and make predictive interpretations of the consequence of the disease variant in a genetic allele. And so that's allowed us some very interesting partnerships where we've understood more and more the power of this automated genome engineering platform, something that's gotten us very, very excited internally. So I'm going to go ahead and wrap up with this slide and a following. I really want to represent to you that we've mastered and understand the translational cycle in terms of utilizing genome engineering to go from target lead through optimization, preclinical validation, clinical trials, and obviously, our GMP clients to date can profess to our ability to get in. So we're really anxious to see these trials move forward, and then obviously supporting analytics associated with approval and production. I just wanted to reinforce with our green and blue bars here that we really have these platform capabilities around both guides and then around cell engineering, that we really do master the combined technologies to execute here. And so I'm going to go ahead and wrap up with a quote here from Jennifer. I mentioned earlier how fortunate we were to have her as an advisor, shaping the direction and a lot of the principles and culture of the company, really driving us to make these new medicines accessible to the people that need them. So with that, I'll go ahead and wrap up. Information in case you'd like to follow up and contact us. Obviously, very happy to be a partner with many others in this community. So thank you very much, and I'm good.